launch the PowerPoint here. Let me get the correct PowerPoint up to launch. Then we'll launch it. There it is. All right, so on Monday, we were talking about protein secondary structure. We talked about beta sheets, and then we talked briefly how to measure tertiary structure. So we went into tertiary structure. Just as a reminder, might as well say that. Primary structure is your sequence of amino acids. Secondary structure is the local backbone conformation. You're talking about your alpha helixes and beta sheets. And now we're talking about tertiary structure. Tertiary structure is simply the overall 3D conformation of one peptide chain, just one chain. That's tertiary structure. And so we talked about how we can find tertiary structure by doing X-ray crystallography. And we also talked about how we can get it using NMR. Now, there's another way, there's a couple other ways, but one of the big ways to get tertiary structure now is through a process called CryoEM, or cryo-electron microscopy. And the idea is that you take your sample and you freeze it very, very fast. So you take your sample and you just dunk it at like minus 196 Celsius incredibly fast. And what happens is that if you freeze water that quickly and that cold, water doesn't have time to become ice. So in ice, water forms the maximum number of hydrogen bonds it can. So water can have 4 hydrogen bonds. And so when you're liquid, just as a review from chapter 2, when you're liquid, you have roughly 3.1 hydrogen bonds, which means 90% of water has 3 hydrogen bonds, 10% has 4 hydrogen bonds. In ice, like everything has 4 hydrogen bonds. Everything's more or less bonded maximally. However, during this cryo-EM, it's frozen so fast and so cold, water does not have time to rearrange into having 4 bonds. It's kind of just like frozen in place. And it's kind of like glass. Now, why that is good is because ice normally destroys proteins. When water rearranges, it will destroy protein in the process. Freezing it this fast doesn't. And so the protein remains in its normal conformation or native state, right? Anytime you see native state, that basically means normal state. This is a state that we see it, inside the cell, your native conformation. And then what you do is you use electrons to image the molecule, right? And so here you can see we have like these little marks. These are GRO-ELs. These are actually proteins, right? And using this technique, one protein that we discovered the structure of is apoferritin, which is this huge molecule. So that's what cryo-EM is generally useful for. It's useful for big molecules that are hard to crystallize, right? That are hard to get through X-ray crystallography. So we can use this technique instead. In fact, this technique 3 years ago now won the Nobel Prize in Chemistry. So any questions about the idea of cryo-EM? Right? So from all this NMR data, all this X-ray data, we have looked at tens of thousands of proteins. And by looking at all these proteins, we've been able to learn certain features about proteins and their amino acids. The features are, one, nonpolar residues are generally on the inside of the protein. So when we talk about the core of folded proteins, a lot of people will say like they're oily. That's because they're just full of nonpolar amino acids. So inside the core of a protein, all your nonpolar amino acids are going to be globbed together. This is because the hydrophobic effect, right? The cell is full of water. Water does not want to interact with hydrophobic or nonpolar amino acids. So it's going to push these together to try and separate them, and that helps a protein fold. So on the inside of a protein are nonpolar amino acids. On the outside, we have charged amino acids, so Arg, His, Lys, Asp, and Glue. Now, it's very, very unfavorable to put our polar amino acids on the inside. If a molecule is charged, it really wants to interact with other molecules. So arginine is positively charged, so it wants to interact with polar molecules and negative molecules. So our charged amino acids are on the outside of the protein. And then our uncharged polar groups, they are usually on the outside because they can hydrogen bond with water. It is possible to be on the inside though, because they can hydrogen bond with either themselves or backbones. So your uncharged polar groups, usually on the outside, but sometimes on the inside, hydrogen bonding. All right, so I do have a question here, and I did make a poll for it. Just so we know our amino acids and our polarity, for A, I give you 4 peptides. I want you to tell me which peptide is most likely to be on the inside of a folded protein. Let's see how well we remember our amino acids. Most likely. It's not important to memorize a negative 196 for CryoEM. It's more important to know that you freeze your protein very fast, very cold. Yeah, I'm never going to try and ask at what temperature is CryoEM carried out at. I'm guessing negative 196 is liquid nitrogen. Let me look that up. Or it might be dry ice, actually. Okay, you know, that is, so negative 196 is the boiling point of liquid nitrogen. So basically you put your protein in liquid nitrogen to freeze it super fast. That's where the negative 196 comes from. All right, let's take a look at this. So 2 is the correct answer, val, iol, lu, trip, alanine. These are your hydrophobic amino acids. 1, you have aspartic acid, asp, so that's not going to want to be in the interior because it's negatively charged. 3, you have a bunch of polar groups. So although there's no charge in there, asparagine, glutamine, and tyrosine to a little extent are somewhat polar. So they could be in the interior, but not as likely as our second protein structure or sequence there. And then third one, we have lysine. Lysine's probably positively charged, so it won't want to be there. So it is 2. Now here's a big picture question. So what would happen to a globular protein? And a globular protein is a protein that has like a hydrophobic core and folds into a 3D conformation. What would happen to a globular protein if you took it out of water and instead put it in a nonpolar solvent like benzene? What would happen if you put a protein into benzene? This is one of those big brain critical thinking questions. Would it denature or turn into a blob? Would it break apart? What is a globular protein again? All right, so a globular protein is a protein that folds into a 3D shape where the center is hydrophobic. All right, so the center is hydrophobic. The outside's polar. And it would denature, right? Because the only thing that pushes these hydrophobic amino acids towards the center is the hydrophobic effect, where water does not want to interact with these hydrophobic molecules, so all of them go right towards the center. So it would unfold if you put it in a nonpolar solute. It wouldn't break apart though. So what would happen is that it would go from some 3D conformation into probably like some random string, right? And so generally what happens is in water, this string folds into a nice 3D shape. But in a nonpolar solvent, there's nothing forcing these hydrophobic amino acids to go towards the center, so it would unfold. But it wouldn't break because it's full of covalent bonds, and so it wouldn't actually break apart, it would just unfold. Unfold and denature are the same thing. If a protein's unfolding, it's denaturing. Those two words mean the same thing. So yes, it would unfold and denature. But yeah, it's the same thing. So our last question here, again, another kind of big brain one. Some proteins travel through the membrane. They are embedded in the membrane. So if here's a protein, right, we have some that are in the membrane. Based on our knowledge of globular proteins on this slide, how would proteins that go through the membrane be different? Like, how would their distribution of amino acids be different? I'm confused on what the question is asking. The question is asking, if you have a protein in the membrane, how are the distributions of amino acids different than in a globular protein? So we know in a globular protein, the middle, the core, is made out of nonpolar amino acids. How would that change if it's going through the membrane? And it looks like somebody got it. So the core wouldn't necessarily be polar. It could inside the membrane. But what would probably happen more likely is that this portion in the membrane would have hydrophobic amino acids on the outside. Now, so we haven't really talked about this. We'll talk about it more about when we talk about membrane proteins, is that usually when you go through the membrane, you do like an alpha helix. That's usually how you do it. Not always, but that is one way you do it. And the way these alpha helixes are made, or what they look like, is that on the outside, you have all these hydrophobic amino acids. So it's really completely different than what we talked about, right? What we talked about is that globular proteins have hydrophobic amino acids on the inside. If you're going through the membrane, you have hydrophobic amino acids on the outside. The extracellular interchannel portions would be hydrophilic. Yeah, so anything that's on the outside of the membrane is hydrophilic. Anything that is like right on the border, right here, it would be hydrophilic. Because just as a reminder, our membranes have a charged head group and a nonpolar tail group, right? That's a membrane. So if we want to be in the membrane, we have to have hydrophobic amino acids interacting with the tail part, hydrophilic amino acids interacting with the head groups. When you said travels, you didn't. So that's why I kind of like clarified it was buried in the membrane, because I realized by using a word like travel, it could mean like you just go through and then you come out the other side. So that's why I wanted to just clarify my question and saying that you're embedded. Yeah, that's why I drew this picture to better get across that. We'll talk about how proteins are different that have to go all the way through the membrane and not be attached. We'll talk about that in a lecture in the future. Questions, though, about the idea of where side chains are located in proteins? So that would be, so the answer is like you would have to have hydrophobic amino acids interacting with the tail, the nonpolar groups. You would have hydrophobic amino acids on the outside, inside the membrane. Yeah. All right. So the last thing about this slide that we're really going to talk are about motifs and what are called super secondary structures. All right. So we talked about secondary structures, alpha helixes and beta sheets. And what happens when you look at, like, you know, we've looked at maybe like 80,000 proteins we have the structures of in our databases, probably more than that. I haven't checked in a long time. Is that you keep seeing the same type of structures over and over and over again, right? And in fact, some proteins, their tertiary structure are just secondary structures. What I mean by that is some proteins, their whole shape is a giant secondary structure. They might just be a giant alpha helix, or they might be what's called a beta alpha beta, right? Where you have beta sheet, alpha helix, beta sheet. Or they could be having a beta hairpin in their structure, where you look at their overall structure and you notice, oh, I have a beta hairpin. Or, you know, a Greek key, which is beta sheets folded up on each other. So I'm not going to ask you to memorize looking at a beta, alpha, beta, beta hairpin. I just want you to be aware of the fact that that when we look at protein structures, it's just not as simple as an alpha helix here, a beta sheet here. Some of these alpha helixes and beta sheets will actually combine into more complex structures. And like I said, some proteins, if they're only made out alpha helixes, they would be called alpha proteins. Some proteins, if they're only made out of beta sheets, they would be beta proteins. And in fact, what we're looking at here is called the immunoglobulin fold. What that is just a beta sandwich that's seen in like antibodies. Like every antibody will have this type of fold. You see it everywhere. The majority of proteins, though, are what we call alpha beta proteins. When we look at a protein's tertiary structure, it's usually made out of both alpha helixes and beta sheets. So that's the majority of proteins, that they have both types of secondary structure. What I want us to be able to do is I want us to be able to look at images of proteins and just be aware, are these proteins alpha proteins, beta proteins, or alpha beta proteins, right? So I have another poll for us. Yeah. So I couldn't figure out how to do Greek letters on Zoom easily. So I want you to look at these images and tell me, is this an alpha protein? Is this a beta protein? Or is this an alpha beta protein? Does it have both? I'll just do a couple seconds more here. 3 is looking like we have some debate on 3. All right. And this, share. So A is an alpha protein. It only has alpha helixes in it. There's no beta sheets. So anytime you see the ribbon, that's an alpha helix. Anytime you see an arrow, that's a beta sheet. There's no like arrows in there, so that's an alpha protein. B, you have both alpha helixes and beta sheets, so that's A, B. C, C was a little tricky to see, right? So if you look in this green, right there and right there, those are alpha helixes. Yeah, kind of tricky to see. So this is a alpha beta protein. There's just not beta sheets in there. There's also alpha helixes. D, that is beta. There are no ribbons. So that is a beta protein. All right, questions about recognizing alpha helixes, beta sheets, alpha protein, alpha beta protein. B is beta as well. B is alpha beta protein. There's alpha helixes and beta sheets. All right. Alpha helix, beta sheet, beta sheet. So it has both. Yes, the arrows do always point to the carboxy termini, right? Yep. So the way the beta sheet is that the flat end of the arrow is the N terminus, the arrow is the C terminus. And so that's how you can determine parallel versus anti-parallel. Yeah, the alpha helix on C was a little bit hidden. There are no cases where the arrow points to the N. It's always going to be pointing to the C termini. The reason for that is because when we write sequences, we always write them N terminus to C terminus. When proteins are made on a ribosome, they're always made N terminus to C terminus. The N terminus is always first. And so to continue with that, we're just saying, you know, the numbering or the direction of the protein is going this way. So it's always N at the butt of the arrow, C at the front end of the arrow. All right. So now when you look at images and you're looking at proteins, now you have a better idea of like what you're looking at: alpha helixes, beta sheets. These spheres are ions or small molecules. So here's that thing's probably iron if I was to guess. All right. So we actually do have one other thing to talk about. Is this idea of domains, right? So a domain is part of the protein that basically folds like as an independent unit. They're almost like if you looked at a large protein, it would have different sections that are all, you know, have their own unique 3D geometry. And they're almost like independent units. They're almost like small independent proteins working together, right? And domains, the structure will usually define its function. And this is something I want to talk about in general. If you want to know what something does, you need to know what it looks like. Structure will always define function, right? If you know what something looks like, you know how it works. So when it comes to domains, to understand what a domain does, we don't really care what the sequence is. We care what it looks like. And here are examples where we have 4 domains. They are made out of different amino acids. Like one has 134, one has 86. So if you were just to compare their sequences, they would be all different. You would think, oh, these parts of the proteins don't relate to each other. However, when you look at their 3D shape, if you squint, they look very identical to each other. So they probably have the same function because they look the same. So when we're talking about what does a domain do, we care about what it looks like. We don't care about the amino acids inside of it. Oh, sorry, there was a question on the previous one that I missed. One sec. So why does D change colors? So the green is probably saying that it's bound to that iron molecule. I'm gonna guess the red means it's super flexible. I would have to look at the original paper to know why they colored things green and red though. Every time you make an image, it's up to you how to color things. So it's either red has some function, or I'm guessing red's very flexible. So they're not sure where it's located 3D. That would be my guess. Anyways, questions about the idea of a domain. Like, what is a domain? What does it do? So remember, domains are like, oh, we do have a question. Domains are critically important for protein functions, right? So a domain might like do a specific reaction, and then you'd find that domain in different proteins, right? So you can say, oh, this protein can also do this function. It's almost like a warehouse, I guess, or an assembly line, where you can think of the assembly line as like one unit, but each little station does something different. Domains are like all part of the same protein, but each domain does something different. On the homework from Monday, I got it wrong. Oh, I don't know what the answer to the homework is without looking at that question again. So I would have to open up the homework. But yeah, domains are super important for structure. For 86, for D, 86 amino acids seem smaller than A. Yeah, it is smaller because it has less amino acids, but it more or less looks the same. That's what we care about. Does it look the same? If yes, they probably do the same thing. So unlike people, for proteins, it's the appearance account. It really doesn't matter what's on the inside. We don't care what the sequence is. All we care about is what it looks like. Proteins are beauty contests. All right, so here are some true and false questions to make sure we just understand that. So let me pull up my little poll again. Here I have actually the questions in there. So let's see if you can get this, these questions to do with domains here. And you can think of motif kind of like a domain. That's what I mean by the word like motif. Does that have a similar motif? That means like, does it have a similar domain? Yeah, motifs and domains are more or less the same thing. A motif just means like, you know, a common element that's, the best way I can put it is that when I say motif, think of it like a domain. That's going to be the easiest way. A lot of words like that I will use interchangeably because a lot of people use in biochemistry, they will use multiple words to describe the same thing, like peptide, protein, peptide chain, all more or less mean the same thing. So yeah. All right, so let's see. Proteins with the same function from a different species are likely to have similar motifs. Yes, that is true. If you have the same function, you're going to have the same kind of like domains to do those functions. So proteins with the same function from different species are likely to be more similar in sequence than in structure. That's false. Again, we don't care what the amino acids are in our domains, in our motifs. We care what is the overall 3D shape. It is the 3D shape that allows us to do chemistry. It is not the actual amino acid sequence. An effective protein motif is likely to be observed in multiple proteins. Yes, if this motif does something really good, if it gets into another protein, right, it's not likely to be mutated away because it's still doing that function. So yeah, we are likely to see the same motif over and over and over again. Proteins with the same motifs will likely form similar functions. Yeah, so that's like the same one as one. If you have the same kind of structure, same domains, same motifs, you're going to do the same exact thing. All right, any questions about that information as I switch my slides? TFTT, yep. True, false, true, true. All right. The last thing we're going to talk about structure-wise, well, general structure-wise, is called quaternary structure. Quaternary structure is multiple proteins coming together to form a giant protein, or multiple peptides coming together to form a giant protein. Here we are looking at hemoglobin. Now we're going to study hemoglobin a lot in this class because it's a very well-structured protein or well-studied protein. But in hemoglobin, we have 4 subunits. A subunit is an individual peptide chain. So peptide chain. So it has an N terminus and it has a C terminus. To make the protein hemoglobin, 4 peptide chains come together to form one protein. So we have one protein made out of 4 peptides, or one protein made out of 4 peptide chains. Now, when these peptides come together, they have certain symmetries that they adopt, because when you're trying to fit together multiple pieces, you have to have some type of geometry restraints to have these fit correctly. So we're just going to briefly go over the bare minimum type of restraints. The first is called cyclic symmetry. Let's say you have a protein made out of 2 subunits. So this is 2 subunits. The only way to make a protein like out of 2 subunits, if those subunits are the same, right? So you have a protein made out of 2 subunits. These subunits are copies of each other. The only way to fit them together is called C2, or cyclic symmetry, 2, C2. And the way to look at this is that this green is your axis of rotation. So if this is subunit 1, subunit 2 is going to be a 180-degree mirror image. Because when it's a 180-degree mirror image, as shown in this image, use image a lot, as shown in this picture, they're going to fit together like puzzle pieces, all right? If your protein is made out of 3 subunits, you're going to have what's called C3, where each protein subunit is separated by 90 degrees. Or sorry, not 90, 120 degrees. So each subunit is a 120-degree mirror of each other. And when you have that, they can fit perfectly. You can have C5 if you have 5 subunits. And it's going to be, what, 360 divided by 5. Oh, what is that? 8, 16, 24, 32, 7, 14. I can't do math right now, 60 degrees. There we go. So in C5, everything is 60-degree mirror image. Now, dihedral symmetry is probably more confusing to think about because in dihedral, you have multiple planes or multiple levels to think about. So in D2, you have 4 subunits, 4 identical subunits, and you have different axes of rotations. So for my first axis of rotation, I'm going to rotate 180 degrees like that. For my second axis of rotation, I'm going to go 180 degrees this way. And unfortunately, I am trying to describe a 3D phenomenon on a 2D plane, so excuse my poor drawings. On our third axis of symmetry, we're going to go 180 degrees this way. And then in our last, we're going to go 180 degrees back to where we started. So we have 4 subunits, but their symmetry is called dihedral. Basically, you have, and I'm going to erase this now, so give me a second. A good way to describe this is a dimer of dimers. So let me write that down. Dimer of dimers. So a dimer means 2 subunits. So you can think about this as these 2 subunits are related through an axis of symmetry. These 2 subunits are related through the same symmetry, and they're more or less stacked on each other. And I think that's easier to see in the D3, in all honesty, where in D3, you have the same type of symmetry as in the C3, right? Where you have that 120 degrees, but it's more or less repeated again. So you have 2 of these. 2 of these symmetry sets. So a D3 is good for 6 subunits. If you have 6 subunits, one way they could interact is through D3. D4 is 8 subunits. I'm not going to try to explain that one because the more subunits there are, the more you have to be good at mental gymnastics and mental picturing. So if any of you who play a lot of video games, you should actually probably be pretty good at this. So one benefit of playing video games, believe it or not, is that you have excellent visual space. You can visualize things in your mind, excellent. And you can rotate things around in your mind visually excellent just by playing video games, because that's what you do when you play a video game, right? So those of you who can think like that can probably visualize what's going on in the D4. Those of you who can't, no worries. I'm not going to give you a complicated picture like this and say, what symmetry is this? Is this a D4? Is this a D5? I'll show you how to do that, though. I'll show you how to work backwards to determine how many subunits we have, what type of symmetry will we have. So that's going to be the question here. Yeah, it's like a giant Rubik's Cube. So yeah, anyone who worked on Rubik's Cubes should be able to figure that out as well, I think. 5 units or 72 degrees. Oh, okay, thank you. Yeah, that's what happens when 5 times 60 is 300. That's what happens when I do math in my head. So let's do this. So let's say I have 4 subunits. What are my possible types of symmetry my protein could have with 4 subunits? I actually have a poll for this. So there's multiple choices here. If I have a protein with 4 subunits, what are all the possible types of symmetry I can have for those subunits? I understand this is like a very confusing concept. But so we're going to just try to keep it simple like this. What are C and D1? C is cyclic, D is dihedral. That's what you mean. Cyclic goes around in a circle. D goes around in a circle, but it does it in 2 different heights. That's more or less what's going on. It's hard. It's very hard to see on 2D. But basically, this is like copied down here. Oh, C1 and D1, that's impossible, right? How can I have symmetry if I don't have a copy, right? So there is no C1, there is no D1. This is talking about multiple subunits coming together. Yeah. Yeah, sorry, I didn't understand the word 1. Usually when you do these, you put the number there. So my mea culpa, my bad. All right. So we're a little bit all over the place here. The correct answer is C4 and D2. All right. So the way this works is that for C symmetries, right, the number is telling you how many subunits are in there, right? C2 has 2 subunits. C3 has 3 subunits. So if I have 4 subunits, you would just arrange them, you know, kind of like that. And they would form a nice whole protein. Or you could be D2, where it's cyclic, cyclic, then above cyclic, cyclic. So D2 or C4. So if I have 6 subunits, based on that, what would be my type of symmetry I could have? What would be my possibilities? And there are others than cyclic and dihedral. You can see on the bottom there, you have T, O, and I. We're not going to touch those because the higher level orders really hurt your mind unless you have like 3D images, which we don't. Even so, they're really hard to think about. Yeah, so if you have 6, you can either be in cyclic 6, C6, or a D3, right? Now, what if I said we're looking at a protein and it has 12 subunits and they're stacked in 2 rings of 6? What type of symmetry is that? That's a D6. Yep. C always goes with number, D always doubles number. More or less, right? So cyclic is whatever number that is. That says how many subunits are in there, and they're just arranged in a circle. D is how many are arranged in a circle, but you have 2 layers, or you have twice that number of subunits in that symmetry. But yeah, multiply the D number by 2, right? But yeah, that is the basics of how multiple subunits come together. If they're identical, they have to have symmetry. Otherwise, they won't fit with each other. I'm trying to think of a good example with my hands. Yeah, let me go to the big screen. So let me share this. So my hands, if I do some symmetry, hey, look, my hands now fit. They fit perfectly. That's basically what a protein's doing, right? If I try to fit my hands like this, they don't quite fit. If I do like that, I still have this part like not fitting to anything. So I need to rotate. And it's not the perfect example because, you know, my hands are mirror images of each other to begin with, but that's the idea. Any questions about that? I know it's a really weird concept. All right, if not, I'll put this up, this video. I will put up a homework, and then we'll continue moving on Friday. So other than that, if you have any questions, as always, please feel free to email me. But if not, I will see you on Friday. Have a good one, everybody.